Hello student, I Dr. R.D. Patil, Head Department of Zoology, Vasantronic Arts, Science and Commerce College, Shada. Dear friends of the SYBSc class student, I teach and discuss about the Zoology course number paper 2, Zoo-302 Biochemistry. In this course, we already discussed about the completion of the regular teaching, learning and notes of the Unit 2. Unit number 1 is carbohydrate metabolism. Unit number 2 is the lipid metabolism. And today we discuss about the unit number 3 is the protein metabolism. In protein metabolism, dear friends, we have already discussed about the first topic with general introduction about the amino acids and the process of the protein synthesis as well as amino acid metabolism which takes place in living animal body. We already discussed about it. Today we discuss general aspects of amino acid metabolism. Dear friends, in protein metabolism, proteins are the polymers of amino acids. Each amino acid contains a central carbon or hydrogen or carboxyl group and amino group. We already discussed this point in first lecture. Major function includes acting as enzyme, receptor transport molecules, regulatory proteins for the gene expression, and so on. General aspects in the protein metabolism, as well as amino acid metabolism, we discuss detail how many processes are creating in the living animal body and how many importance of the amino acid for the living animal body. The protein is nothing but the building blocks of the amino acids. Amino acids are most important in a living animal body. We also in the form of the essential amino acids or non-essential amino acids. We have the process is generally takes place. We discuss in first lecture amino acid pool sources and utilization. Today we discuss the general aspects of amino acid and metabolism. In this process, the amino acids obtained from dietary source or body protein turnover are utilized for protein biosynthesis and the production of a wide range of nitrogen containing compounds. Dear friends, the amino acids undergo certain common reactions like transamination followed by deamination for the liberation of ammonia. The amino group of the amino acids is utilized for the formation of urea, which is an auxiliary end product of protein metabolism. Means in our amino acid metabolism, protein biosynthesis and production is the most important to as required by the living animal functions. The carbon skeleton of the amino acid is first converted to keto acids by transamination, which meets one or more of the following fates. So this process or general aspects of the amino acid metabolism also takes place the following fates. There are the four stages. Out of these four, this Roman first is the utilized to generate energy. Second is used for the synthesis of glucose. Third, diverted for the formation of fat or ketone bodies, and inverted in the production of non-essential amino acids. So these four stages, general pictures of amino acid metabolism is shown in figure 3.2. This figure we also schematic representation is denoted by in this schematic representation. That is a dietary protein which are coming from body proteins and protein synthesis takes place. These amino acids is carried on by the transamination process to form the keto acids. At the time the transamination alpha ketoglutarate, where also specific enzymes are utilized to convert glutamate is also deamination, NH3 ammonia and urea is another also called the urea cycles. And next keto acids which are also formed by the energy glucose, fat, and non-essential amino acids. This is utilized in the next reactions of the an overview of the amino acid metabolism. In this schematic representation is a show this diagram. Next is the biosynthesis of amino acids. In biosynthesis of amino acids, entry of nitrogen into living system. First, as carbon and oxygen enters into composition of molecules through CO2 and hydrogen enters through water. Nitrogen enters through biomolecules through ammonia and nitrates. Certain bacteria reduce atmospheric nitrogen to ammonia by a process called nitrogen fixation. Most bacteria and fungi and all plants obtain nitrate NO3- from soil and reduce it ammonia. So this specific process takes place in the as by the biosynthesis of amino acids to reduce this. All specific processes are also all ammonia is then made to react with alpha ketoglutarate to synthesize the amino acids glutamic acid and glutamine. These amino acids then serve as the donors of amino groups for synthesis of the other amino acids for which the carbon hydrogen skeleton are derived from the different intermediate of glucose catabolism. Means, dear friends, in this process is most important the amino acids in different forms or different groups are most important to conducting the specific process of the biosynthesis of the amino acids. All ammonia is then made to react with as specifically formed by the these processes of the catabolism. The various amino acids then serve to not only as building blocks of protein but also as donors of amino acids donors of amino groups to a variety of nitrogens containing biomolecules such as the nitrogenous bases of nucleotides, that is building blocks of nucleic acid, DNA and RNA, deoxyribonucleic acid and ribonucleic acid, uh, neurotransmitters, hormones, etc. Thus, the route by which nitrogen of the nature enters into the chemistry of life is atmospheric N2 or soil nitrate ammonia, uh, glutamate and glutamine is the specific to convert the specific process for the biosynthesis of the amino acids. In the process that is the specifically these processes occurs only in microbes, fungi and plants. These organism can synthesize all the 20 amino acids. Human and other mammals, however, can synthesize only 10 non-essential amino acids and obtain the 10 essential amino acids from the food. Means in your living animal body, the uh, amino acid 20 amino acids are formed. Out of these 20, only 10 amino acids are formed by the living animal body and 10 essential amino acids are from the food. According to a nutritional scientist, the nutritionally essential amino acids are termed essential or indispensable amino acid, and the nutritionally non-essential amino acids are termed non-essential or dispensable amino acids. These are two groups of the amino acids. Nutritionally non-essential amino acids are more important to a cell than the nutritionally essential ones. The essential amino acids are methionine, arginine, tryptophan, valine, leucine, isoleucine, phenylene, threonine, lysine, and histidine. Whereas the non-essential amino acids are alanine, glutamic acid, aspartic acid, glutamine, asparagine, serine, glycine, proline, cysteine, and tyrosine. Essential amino acids are defined as one which cannot synthesize by organism from substances obtained. These substances are specifically obtained to as ordinarily present in the diet at the rate of physiological requirement. Biosynthesis of both essential and non-essential amino acid from different precursors or sources is described below as a specifically process which takes place in the form of, that is, the A process, biosynthesis of non-essential amino acids. This is the most of non-essential amino acids formed from amphibolic intermediate as follows. Means this biosynthesis of non-essential amino acid is the mostly to as a following of this amino acid. That is, first is the alanine in this group. Second is the glutamic acid. Third, glutamine, aspartic acid, asparagine, serine. Next is the serine, glycine, proline, cysteine, and tyrosine. We already discussed about the. It is the also called as a non-essential of the amino acids. And next is the essential amino acids. This is the second is the biosynthesis of essential amino acids. Nutritionally essential amino acids are synthesized by bacteria, but the synthesis does not take place in mammalian tissue following amino acid biosynthesis. That is arginine, valine, histidine, leucine. Next is the leucine, isoleucine, lysine, methionine, phenylalanine, threonine, and tryptophan. This is the essential amino acids which are second steps to biosynthesis in the specific processes, that is the essential amino acids. We have the specifically these groups, that is alanine. It is formed in a first process, that is biosynthesis of non-essential amino acids, that is alanine. It is formed from pyruvate by transamination in presence of coenzyme pyridoxal phosphate B6PO4 in all forms of the life. Second is the glutamic acid. In all forms of life, glutamic acid is formed from alpha ketoglutarate by glutamate dehydrogenase. Bacteria contain only an NAD plus dependent dehydrogenase, whereas yeast and fungi contain two glutamate dehydrogenases specific for NAD and for NADP. In third, glutamine in plants and animal and bacteria. Glutamine is synthesized from glutamate by glutamine synthesis NS4 plus aminate glutamate. So this specific reaction takes place as a schematic representation which takes place in the notes. Next is the aspartic acid, also is formed by transamination or oxaloacetate. Asparagine is also synthesized from separate by the asparagine synthesis. ATP and Mg+2 are also required these reactions. Next is the serine. In mammalian tissues, serine is synthesized from 3-phosphoglycerate and intermediate of glycolysis by two pathways. One pathway uses phosphorylated intermediates and the other uses non-phosphorylated intermediate. It is a majority of the serine is synthesized by the pathway via phosphorylated intermediate. Plants and microorganisms follow this path as specifically this takes place in schematic representation. Next is the glycine. Glycine is a synthesized from the serine as well as proline. Next is the proline. It is synthesized from glutamate or alpha keto glutarate by reversal of reaction for protein catabolism. Next is the cysteine, is also formed from methionine. Methionine is the first converted to the homocysteine, which is converted to cysteine in conjugation with serine as shown below in specific schematic representation. Tyrosine also is conversion of phenylalanine to tyrosine is catalyzed. specifically most important to tyrosine and phenylalanine are biosynthesized from pre-phenate, which is converted into amino acid specific intermediate. So in this way, this is a formed by the non-essential amino acid or biosynthesis of non-essential amino acid. Next is the B, biosynthesis of essential amino acid. It is most important to these 10 amino acids which are uh, mainly not formed by the animal bodies. In living animal body is produced or creating to extra enveloping by the food or any plant materials are eating. In this way, the human body has produced or formed by this 20 amino acid. It is called essential amino acid. This is arginine, is synthesized from alpha ketonic and later transmitted. Next is the valine, is produced by a 4-enzyme pathway. Histidine, the synthesis of histidine in E. coli is a complex pathway involving. And leucine, uh, this is also synthesis pathway diverges from the valine pathway beginning with alpha isovalerate and alpha isopropylamate synthesis catalyze this condensation with acetyl coenzyme to produce alpha isopropylamate. So this specific process is converts in isomeric processes. Next is the isoleucine. Uh, in plants and microorganisms, isoleucine is biosynthesized from pyruvic acid and alpha ketone. Next, lysine is synthesized from aspartate via the dinoplates. Also, as a pathway, the initial two stages of the DAP pathway are catalyzed by uh, aspartokinase and apparate to semialdehyde dehydrogenase. Next is that 7-methionine. Biosynthesis of methionine by the transsulfation pathway starts with aspartic acid. Relevant enzymes include. Next, 8 is the phenylalanine is biosynthesized from pre-phenate, which is converted to an amino acid specific intermediate. This process is mediated by the phenylalanine. Next is the threonine. In plants and microorganisms, threonine is synthesized from aspartic acid via alpha asparatyl semialdehyde and homoserine. Homoserine undergoes O phosphorylation. This group, O phosphorylation. This phosphate ester undergoes hydrolysis concomitant with reaction of the OH group. And last is the tryptophan. Is biosynthesis involves conversion of chlorismate to anthralinate using anthralinate synthesis. This enzyme requires either glutamine as the amino group donor or ammonia itself. So in this specific processes, this metabolism of or biosynthesis of essential amino acid as well as biosynthesis of non-essential amino acid. In the two processes, that is capital A is the biosynthesis of non-essential amino acid and capital B is the biosynthesis of essential amino acid. Is most important in the protein metabolism processes. So in this way, this is the next, that is a transamination and specific processes, that is the mechanism of transamination, deamination, also as a metabolism of ammonia. Next is the decarboxylation, and last is the most important as the specifically the urea cycle. This process are completely discussed about in the next chapter in this way. So briefly, some schematic slides as well as a representation to takes place in the U. There is running as a briefly to discuss the U. That is a biosynthesis of amino acid. In this way, these all kinds of the processes it takes place. That is amino acid classification, some definition to nitrogen balance, protein requirements, biological value, digestion of protein, absorption of protein, metabolic rate of the protein metabolism of amino acid, as also all kinds of the processes, removal of the ammonia by deamination, transamination, and trans deamination, fate of carbon skeletal of amino acid, metabolism of ammonia, all kinds of processes are takes place in the protein metabolism chapters. That is this we already discussed about the What is metabolism? Total all Total of all chemical changes that occur in body includes anabolism and catabolism. Anabolism is a processes in which energy requiring processes where small molecules join to form large molecules. Example, glucose plus glucose. In catabolism, energy releasing processes where large molecules broken down. Means that in simple way. In catabolism, in anabolism, energies are producing. In anabolism, producing energy are utilized. So energy in the carbohydrate, lipids, proteins is used to produce ATP, adenine triphosphate, through oxidation reduction reactions are takes place these processes. In the next step, that is a metabolic pathway of the enzymatic reactions of the metabolism from a network of the interconnected chemical reaction of pathway. This molecular pathway are called intermediate because the products of the one reaction become the substance of the next. Enzyme control the flow of energy through a pathway and takes place as a intermediary metabolism in the schematic repetition. In the oxidation reduction reaction occurs via the loss of hydrogen or the gain of oxygen, where whenever one substance is oxidized, another substance is reduced. Oxidized substances loss energy, reduced substances gain energy. That is actually is also process which are the same for the anabolism or catabolism. Coenzyme acts as a hydrogen or electron acceptors. Two important coenzymes are nicotinamide adenine dinucleotide NAD and flavin adenine dinucleotide FAD as the most important products to get by the energy by living organism. So these stages of the metabolism, energy containing the nutrients processed and three major part, that is digestion, anabolism, and formation of catabolic intermediate, whereas the nutrients are and are broken down by the catabolic pathway, which takes place by the oxidative breakdown of this reaction in schematic representation are seen. Protein metabolism, non-essential amino acid can be formed. Transamination transfer of an amine group to keto acid can also be eaten. It is used for energy. Amino acids undergo oxidative deamination. Ammonia and keto acids are produced as byproducts of oxidative deamination. Ammonia is converted to urea and excreted. Amino acids are not stored in the body. So this is a specific process we already discussed about in the briefly in before to description are discussed in specific processes. Next is the this is a protein catabolism reactions are takes place in the schematic representation. Briefly, to have the metabolism of protein, metabolism amino acids, nitrogen is enter the body. Dietary proteins cannot be stored as such, but used for formation of tissue proteins due to there is a continuous breakdown of endogenous tissue proteins or the takes place in the specific reactions. So this is a essential amino acid, non-essential amino acid. We all kinds of the already discussed in separately as the slides. To that is a 10 amino acids are essential. Then later on the next 10 is a non-essential. So all kinds of the amino acid meaning or category, that is the histidine and arginine are the semi-essential. They are essential only for infants growth, but not for old children or adults. Where in adult histidine requirement is obtained by intestinal flora and arginine by the urea cycle for formation of new tissue protein. So this is the requirements of the essential and non-essential amino acid for the reaction of the protein metabolism. Dear friends, we nitrogen balance, it is NB, is a comparison between nitrogen intake in the form of dietary protein and nitrogen loss as undigested protein in feces, NPN, as urea, ammonia, creatine, and uric acid in urine, sweat, and saliva, and losses by hair, nail, skin. NB is important in defining overall protein metabolism of an individual nutritional nitrogen requirements. So it is called the nitrogen balance. So in short, it is NB. It is most important for the every living animals to maintaining the undigested or digested proteins in feces is also required as a dietary proteins. Three states are known for NB. Normal adults, positive nitrogen balance negative nitrogen balance, as specifically reactions are takes place in the protein requirements for the humans in healthy and disease conditions are to required by the specifically 0.8 kg body weight day. That requirements is increased in the healthy condition during the periods of the rapid growth, pregnancy, lactation, and all those sense. Protein requirement is increased in disease states, illness, major trauma, and surgery. Requirements for the proteins should be reduced in hepatic failure and renal failure. It is a specifically healthy and disease condition. The requirements of proteins are most important. Dear friends, biological value of the protein also discuss in specific way as a digestion proteins. We also most important in endocrinologists. All kinds of the requirements are specifically as a briefly process or takes place in next units as a pepsin or renin. That is a gastric phase of the protein digestion is most important. This is a pancreatic phase of the protein digestion in this schematic representation. That is a intestinal phase of protein digestion. Absorption of amino acid digestion. All kinds of the processes are really to discuss in the next chapters, which are briefly to schematic representation. So this is a specifically this metabolic phase of the amino acids later on discuss in the next units. We detail discuss about the specifically this transamination and this process are detailed or briefly discuss to alternately in the next lecture. So dear friends, we discuss in briefly. Thanks. Stay home, stay safe.